and the ubiquitin proteasome system metabolically removes about 80% of these proteins in eukaryotic cells. So how the ubiquitin proteasome system works is, if you look at the image on the right, you have these round ubiquitin molecules, the blue spheres that you can see, which covalently attach to a protein of interest. And once they're finished attaching to the protein of interest, the ubiquitin signals for protein degradation by the proteasome. Now, looking at the image to the left at the proteasome consists of two major subunits, and each subunit has its own functionality. And the RP subunit is responsible for recognizing ubiquitin and the protein, as well as removing ubiquitin once the protein is engaged by the proteasome. Furthermore, it unfolds the protein and translocates it into the CP, or the core particle, where the protein is degraded into peptide product. Next slide, please. Much of our understanding on proteasomal degradation has been based on terminally initiated proteins. So looking at the image on the left, you see the orange sphere is a protein, and then coming from a sphere is an unstructured region. And the region, the terminal end of the region of the protein starts at the triple ATP ACE motor, and that motor mechanically unfolds the protein by ATP hydrolysis. Now, there are three major factors that lead to successive protein degradation, but the main one is proper stability kinetics for proteasomal unfolding. And just a general insight, the motor, the AAA motor, efficiently unfolds unstable domains of proteins than stable domains of proteins. So what we know based on terminally initiated protein degradation transcends to my objective on the right, where you see internally initiated protein. So what I mean is, if you notice, you see an internal loop that's entering into the proteasome. So we want to know, based on this hypothesis, what type of products are we getting made from proteasomal degradation? Do we get, in the first image on the bottom, the proteasome have problems with engaging the protein and cleaves, causing both domains to escape? Or do we get, in the second image, the proteasome completely degrades the protein? Or in the third image, which is based on our hypothesis, based on the stability kinetics, does the proteasome degrade the more unstable domain than the stable domain? So if you're interested in learning more about internally initiated protein degradation, I welcome you to come and view my poster. Thank you so much.